Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullah. This is our 10th lecture for the immunology course, and it's going to be about the cytokines. So the objectives of this lecture are going to be define and know the general properties of cytokines, classify cytokines, know the number of cytokine receptors, and the types of the cytokine receptors, what are the biological functions of the cytokines, and if the cytokines are used to treat diseases or illnesses, and what types of cytokines treat which type of disease. So to start off, what are cytokines? Cytokines are a group of low molecular weight polypeptides or proteins which are secreted by activated immunocytes or some matrix cells and possess high activity and various function. So it is used to describe a diverse group of soluble proteins and peptides which act as a humoral regulator at nano to picomolar concentrations. Their major function are to mediate and regulate immune response and inflammatory reactions. So they are low molecular weight polypeptides released from cells to affect other cells and mediate signaling between the cells. So most cytokines are low molecular weight polypeptides or glycoproteins, and most of them are monomers. Natural cytokines are secreted by activated cells such as the activated immune cells, matrix cells, and tumor cells. One kind of cytokines can be produced by different cells. One kind of cells can secrete different cytokines. So one cell can secrete different types of cytokines, and one kind of cytokine can be produced by different cells. Cytokines initiate their action by binding to specific membrane receptor on target cells. So they are released from certain cell, and then they initiate their action by binding to their receptor on the target cell. Cytokines can act on the cells that produce them, and that's what we called previously autocrine. So it's released from this cell, and then it binds to its equivalent receptor on the same cell that it was released from to have the activation effect, and this is called autocrine. On other cells in immediate or other cells in the immediate vicinity within the cell, this is called paracrine. So it's released from cell X to target cell Y or neighboring cell Y, and this is paracrine. Or on cells at distance, and this is called endocrine after being carried in blood or tissue fluid. So we have here the inducing stimulus. It's going to activate the cytokine gene. The cytokine is going to be produced by this cell, and then it is going to affect the neighboring cells by binding to the receptor on the neighboring cell. This is going to lead to gene activation of the cytokine, and this is going to give the end result or the biological effect of the cytokine released. So autocrine, it's released from this cell, and then it binds to the receptor on the same cell to activate the cytokine gene. Paracrine released from this cell to the neighboring cell, and endocrine, it goes through the circulation to distant cell to bind to the to its equivalent receptor on the distant cell and activate the cytokine gene. So we have like 6 types of cytokines. We have the interleukins, we have the lymphokines, the monokines, the interferons, the colony stimulating factor, and the chemokines. Interleukins produced exclusively by leukocytes, lymphokines produced by lymphocytes, monokines, they are produced by monocytes, interferons involved in antiviral response, colony stimulating factor, it supports the growth of cells in the semi-solid media and has to do with the hematopoietic differentiation. Also, we have the chemokines, which promote chemotaxis, and they are responsible for attracting the blood cells to the location of infection. So what are the effects of cytokines? We have pleiotropism, refers to the ability of one cytokine having multiple effects on diverse cell types. Redundancy refers to the property of multiple cytokine having the same or overlapping functional effects. Synergy refers to the property of two or more cytokines having greater than additive effect. And antagonism refers to the ability of one cytokine inhibiting the action of another. And in a previous lecture, we had a slide where these were mentioned: pleiotropism, redundancy, synergy, and antagonism. So here we can see the pleiotropism effect. So the activated CT4 helper cell is going to release interleukin-2, which is going to lead to differentiation of both the IgE and T helper 2 differentiation. Redundancy, as we can see, interleukin-2 and interleukin-4 has the same role in proliferating the B cell. So two types of cytokine doing the same work. Synergy, here we can see that interferon gamma and tumor necrosis factor increase expression of class 1 MHC molecules on many cell types, so they have more than additive effects. Antagonism, so the interferon gamma leads to macrophage activation, while the interleukin-10 inhibits macrophage activation. Cytokine general actions. We have multiple actions for the cytokines. Development of cellular and the humoral immune response. They play an important role in the development of the cellular and the humoral immune response. Induction of inflammation, regulation of hematopoiesis, control of cellular proliferation and differentiation, induction of wound healing, and also they aid in chemotaxis. So classification of cytokines, we have the interleukins, interferons, tumor necrosis factor, colony stimulating factor, chemokine and transforming growth factor. And in the next slides, we are going to talk in some detail about each and every one of those types of cytokines. So the interleukins. Family, we have the interleukin-1 family, which is typically secreted by very early in the immune response by dendritic cells and monocytes or macrophages. So among the early response is the interleukin-1, which is secreted by the antigen-presenting cells. Its secretion is stimulated by recognition of viral, parasitic, or bacterial antigen by innate immune receptor. So once a foreign antigen or foreign pathogen is detected by the innate immune system, this is going to activate and lead to the production of interleukin-1. Interleukin-1 family members are generally pro-inflammatory. They are pro-inflammatory. What does that mean? In the next slide, we're going to answer that. So they induce an increase in the capillary permeability at the site of cytokine secretion. Increased capillary permeability, amplification of the level of leukocyte migration into the infected tissue, and has a systemic whole body effect and signals the liver to produce acute phase reactant and interleukin-6. These proteins further induce multiple protective effects, including the destruction of viral RNA. So the goal of releasing the interleukin-1 is destruction of the viral RNA or the invading pathogen via increasing capillary permeability, amplification the level of leukocyte migration into the site of infection, and signal the level to produce the acute phase reactants such as C reactive protein and interleukin 6. Generation of a systemic fever response, which helps to eliminate many temperature sensitive bacterial strains. So it helps in production of fever, and this elevation works as a protective mechanism because some bacterial strains cannot grow in elevated temperatures. By the way, who knows the mechanism of fever? It has to do with the hypothalamus, it has to do with the heat receptors and cold receptors. This is a homework for you. روحوا شوفوها دوروا عليها كيف بتصير. Interleukin 18 stimulates production of the macrophage activating cytokine interferon gamma by natural killer cells and T cells. Also among the interleukin 1 family, we have the interleukin 18, which stimulates the production of macrophage activating cytokine interferon by natural killer cells and the T cells. In the innate immune response, interleukin 1 also activates both T and B cells at the induction of the adaptive immune response. زي ما حكينا بالبداية بس عندنا detection of pathogen by the innate immunity. Interleukin 1 is going to be released to activate both the B and T cells. So here we can see the local inflammation on the endothelial level. It's going to increase the permeability. It's going to lead to upregulation of the adhesion molecule on the inside of the vessels in order to lead to leukocyte rolling and extravasation. Also, the interleukin 1 and tumor necrosis factor is going to activate the leukocytes, and it's going to lead to production of the chemokines to attract the white blood cells to the site of infection. We have systemic protective effect in the brain. As we said, it induces fever by a mechanism اللي هي طلبنا منكم تدوروا عليها الميكانيزم كيف بتصير, induced by the TNF, interleukin 1, interleukin 6. Also, it has an effect on the liver. Interleukin 1, interleukin 6 lead to acute phase proteins اللي هو C reactive protein or the CRP. Also on the bone marrow, it induces leukocyte production by TNF, interleukin 1 and interleukin 6. Also, the interleukins might have systemic pathological effects, but we're not talking here about the interleukin 1 family. Here we're talking about another family, which is the tumor necrosis factor family, and these effects include decreasing cardiac function and thrombosis, leads to formation of thrombus, and it also leads to metabolic abnormalities due to insulin resistance. We're still talking about the interleukin family. We have the hematopoietin family, which is classified or called as class 1 because it was the earliest to be structurally categorized, not first to be discovered. هلا راح نشوف إنه الانترفيرون they were the first to be discovered. But the first to be structurally characterized are the hematopoietin family. A large family of small cytokine molecules with functional diversity. Not all are involved in hematopoietic function. So their cellular origin and target cells are as diverse as their ultimate function, which range from signaling the onset of T and B cell proliferation. Here we're talking about interleukin 2. Interleukin 2 function in T and B cell proliferation, signaling the onset of B cell differentiation to plasma cell and antibody secretion. Here we're talking about interleukin 6. signaling the differentiation of a T helper cell along one particular differentiation pathway versus another. We have the interleukin 4 and the interleukin 12, which determines the differentiation, which it either takes this direction or this direction of the T helper cell. And finally, initiating the differentiation of particular leukocyte lineages, such as the granulocyte monocyte colony stimulating factor, the GM-CSF, or the granulocyte colony stimulating factor. And these are examples of some of the class 1 members of the cytokines. We have the interleukin 17 family, which is the most recently described cytokine. It has a pro-inflammatory cytokine effect, and we have the interleukin 17A, B, C, D, and F. Signaling through most members of this family culminates in the generation of inflammation. So interleukin 17 has a role in generation of inflammation. It's released by activated T cells and stimulates the production of factors that signal a pro-inflammatory state, including interleukin 6, chemokines XCL8, and granulocyte colony stimulating factor. So interleukin 17 family plays a role in the generation of inflammation and attracting cells to the location of inflammation. Second type of cytokines, we have the interferons, which is classified as class 2. It was the first cytokine to be discovered, as we said. So we have three types of interferons. We have interferon alpha, beta, and gamma. Type 1 secreted by not only macrophage and dendritic cells, but by also virus-infected cells. So we have the interferon alpha and interferon beta. Type 2 produced by activated T cells and natural killer cells, known as interferon gamma, and cytokines include interleukin 10. So here, as we can see, interferon alpha, it's type 1, produced by leukocyte, and the main function is antivirus immune regulation. It protects against viruses and has a role in immune regulation. Interferon beta, type 1, produced by fibroblast, and it is anti-cancerous. Interferon gamma, classified with type 3, and it's produced by T helper 1 and natural killer cell. It has a weak antiviral effect, stronger immune regulation effect, and anti-tumor. So when we talk about interferon that works against viruses, we're talking about interferon alpha mostly. Interferon gamma is used medically to bias the adaptive immune system to the cytotoxic response in diseases such as leprosy and toxoplasmosis. What does that mean? معناته إذا أعطينا interferon gamma معناته إحنا قاعدين بنحفز الجسم to produce more cytotoxic T cells, and so these cytotoxic T cells to target cells which are infected by intracellular pathogens to kill these cells. And in this case, antibody response are less effective because we're talking about intracellular infection. Antibody response is good enough when the invading pathogen is still outside the cell. Once it's intracellular, then we need something to induce a strong cytotoxic effect. Interferon gamma does that. And it induces the activation of macrophages with subsequent destruction of any intracellular pathogen and differentiation of cytotoxic T cells. All three interferons, alpha, beta, and gamma, increase the expression of MHC complex proteins on the surface of cells, thus enhancing their antigen presentation capability. So they increase the ability of all the cells to express major histocompatibility complex in order to present the intracellular pathogen or organism or foreign antigen which is intracellularly into the surface of the cell, so that can be recognized and targeted by the cytotoxic T cell. So here we see the mechanism of induction of type 1 interferon by the viruses. Here we have the virus. It's been internalized into the endosome, and then the DNA or RNA of the virus binds to the or what we call the PAM. It binds to the pattern recognition receptor. Then it's going to activate a signaling pathway, which is going in the nucleus to activate the production of interferon alpha and beta. Once the interferon is activated and released from this viral infected cell, it's going to bind to the receptor on the uninfected cell, and this is going to lead to phosphorylation of translation initiation factor, elongation factor 2 alpha, activation of RNAs, and this is going to lead to inhibition of viral protein synthesis, degradation of viral RNA, and inhibition of viral gene expression and virion assembly. So the net effect of the interferon gamma is affecting the production of the viral protein, and that's how it inhibits the viral replication. The third type of cytokine family is the tumor necrosis factor family. Can signal development, activation, or death of certain cells, which induces apoptosis or programmed cell death. Tumor necrosis family, tumor necrosis factor. From its name, it leads to necrosis, where the cell dies by apoptosis. The mechanism of cell death, in which the cell dies from within it, is fragmented into membrane-bound vesicles that can be rapidly phagocytosed by neighboring macrophages. So there's fragmentation and then phagocytosis by neighboring macrophages. Cytokines of the tumor necrosis factor family, there are two members having the same name of the TNF family. We have the TNF alpha, TNF beta. Also, we have the FASL or the FAS ligand member of the TNF family. And the TNF alpha and beta, both of these are secreted as soluble proteins. TNF alpha is pro-inflammatory cytokine produced primarily by activated macrophages and lymphocytes in response to infection or inflammation. It's pro-inflammatory, and it's produced by macrophages and lymphocytes. TNF beta is produced by activated lymphocytes and deliver a variety of signals on binding to neutrophils in endothelial cells, lead to increased expression of MHC and of adhesion molecule. So TNF, as we've seen previously, plays a role, TNF, with interleukin-1 in the cellular, in the leukocyte extravasation and drolling, where the neutrophils bind to the endothelial cells, leading to increased expression of MHC and adhesion molecules. Also, we have the FAS ligand, which regulates immune system and leads to progression of cancer. And this member, as other members, lead to apoptosis induction. The fourth type is the colony stimulating factor. Colony stimulating factor cytokines stimulate proliferation or differentiation of pluripotent hematopoietic stem cell and differentiation progenitors. So it has to do with hematopoiesis. We have the multi-CSF or the interleukin-3, we have the granulocyte macrophage colony stimulating factor, we have the monocyte colony stimulating factor, we have the granulocyte colony stimulating factor, also we have the stem cell factor, and also we have the erythropoietin or the EPO, which is secreted by the kidney in response to cellular hypoxia, and the net result is production of more red blood cells to cope with this hypoxic status. Hematopoietic stem cell differentiation pathway and lineage specific markers. So we can see here the interleukin 3, granulocyte macrophage colony stimulating factor, monocyte colony stimulating factor roles in the differentiation of the different steps of the hematopoietic stem cell. Here we can see the interleukin, the SCF. Here we can the interleukin 3, the SCF. Here the granulocyte macrophage colony stimulating factor. Here the monocyte colony stimulating factor, granulocyte monocyte colony stimulating factor. So they play an important role in almost all the steps of differentiation of the hematopoietic cells, as we can see in this figure. The fifth type of cytokine is the chemokine, which directs the migration of leukocytes through the body. So it directs the leukocytes through the body to the site of infection and inflammation. They are a structurally related family of small cytokines that bind to cell surface receptor and induce the movement of leukocyte up toward the chemokine source. This soluble factor-directed cell movement is known as chemotaxis. Even chemokines play a role in chemotaxis, and once they are released, they attract the white blood cells, the leukocytes, into the site of inflammation. So we have the CXCL group, which attracts neutrophils. We have the CCL group, which attracts monocytes and macrophages. CXCL attracts neutrophils, and the CCL attracts monocytes and macrophages. Here, this is just to show the role of the TNF, the interleukin-1, and other cytokines in the leukocyte extravasation steps that we've talked about previously. The sixth type of cytokine is a transforming growth factor. From its name, are cytokines which stimulate the growth of their target cells. So we have the transforming growth factor beta, epithelia growth factor, which plays a role in stimulating the growth of epithelium. Vascular endothelial cell growth factor stimulates the growth of the vascular endothelium, and the fibroblastic growth factor, which stimulate the growth of the fibroblasts. So that's about all the families of the cytokines. Now we're going to talk briefly about the types of cytokine receptors. We've already enumerated these types in previous lectures. So we have the membrane-binding cytokine receptor. The receptor consists of extracellular region, transmembrane region, and cytoplasmic region. We have the extracellular, we have the transmembrane, and we have the cytoplasmic region for these cytokine receptors. Can be grouped into 5 families according to structure and function. So we have the Ig receptor superfamily, we have the type 1 CK receptor superfamily, type 2 CK receptor superfamily, type 3 CK receptor superfamily, and the G protein linked receptor superfamilies. So if we classify cytokines into functional categories, we can classify them into three main functional categories. The one which mediate or regulate the innate immunity, the one which mediate and regulate the adaptive immunity, and the one which stimulate hematopoiesis. So for the one which regulate the innate immunity, we have the TNF, the interleukin-1, interleukin-12, interferon type 1 and interleukin 10. Mediate or regulate the adaptive immunity, we have the interleukin 2, 4, interferon gamma, and tumor growth factor beta. Stimulate hematopoiesis, we have interleukin 3, interleukin 7, GM-CSF and M-CSF, and the SCF factors as well. So for the specific interleukins functions, so if we want to talk about individual interleukins, we have the interleukin 1, which plays a role in the inflammation. Interleukin 2, growth factor for B and T cell, leads to clonal expansion. Interleukin 3 has a role in hematopoietic growth factor, stimulates colony formation of blood cells. Interleukin 4 stimulates development of T helper 2 cells from naive T helper cell. So interleukin 4 stimulates development of T helper 2 from the naive T helper cell. It takes the T helper cell from the T helper 0. If the T helper 0 is acted upon with the interleukin 4, it's going to differentiate into T helper 2 cell. It stimulates the IgE class switch from IgG1 to IgE in the case of allergy. Interleukin 5 produced by T helper 2 cell and aids in the growth and differentiation of eosinophils. Interleukin 6 is an acute phase reactant which leads to the production of and the action on liver as well to produce the C-reactive protein as well. Interleukin 10 has a suppressive or inhibitory effect of the inflammatory response and inhibits production of the interferon gamma, interleukin 2, interleukin 3, tumor necrosis factor alpha, and the GM-CSF. And the interleukin 12 is involved in differentiation of the naive T cell into T helper cell. So the naive T cell, if it's acted upon with interleukin 4, it's going to lead to development T helper 2. If it's acted upon by the interleukin 12, it's going to lead to differentiation into T helper 1 cell. So are there any implications or are there any application for cytokines in treatment of patients? These are some of the clinical applications of the cytokines. So cytokines and cytokine inhibitors can be used in many clinical applications and treatments. Advantages, we have a known ligands for the cytokines. We have receptors and the mechanism of action is already known for those cytokines. The problem with cytokine therapy is the effective dose level. The effective dose level is high, so we need to give high doses. Short half-life. It has a short half-life also and can cause unpredictable side effect. What are the some of the applications? So we have the colony stimulating factor. It helps in hematological disorders associated with cancer therapy. The erythropoietin, the EPO, has to do with treatment of anemia associated with kidney disease. Interferon alpha has a role in as an antiviral therapy in hepatitis B and C and in multiple sclerosis. Interferon beta has a role in multiple sclerosis. Interferon gamma has to do with chronic granulomatous disease. Interleukin 2 is an FDA approved treatment modality for treatment of kidney cancer and melanoma. And we have interleukin 11, which has to do with thrombocytopenia in cancer patients. It's used to treat thrombocytopenia in cancer patient. So that's all for the cytokines lecture. Just I have another homework for you. In this pandemic, the SARS-CoV-2 or the COVID-19 pandemic, نحكى كثير عن السيتوكاينز ونحكى كثير عن إشي اسمه cytokine storm واتسلط الضوء عليه لفترة لا بأس فيها ونحكى عن السيتوكاين ستورم وعلاقته بالprogression of the rapid progression and worsening of the condition of the COVID-19 patients and death. مطلوب منكم انه تعرفوا لي وتدوروا لي شو هو السيتوكاين ستورم وما علاقته بالسارس كوفي 2 infection. ممكن يجي عليه سؤال في الامتحان. That's all for this lecture. Thank you for listening.